I hope you guys have enjoyed Seattle. This is my now hometown. I've been here over 20 years. This is a little more typical May weather for us, so we shut down the sun for a little while. We can only have a few days of it in Seattle before we get just a little twitchy. So I'm going to talk to you today. Oh, and the other thing I want to say is Happy Mother's Day to all the mothers out there. I didn't put in a slide, but you guys rock. Yeah. And it's great to be here with you today for that. So as Tracy said, I'm going to talk to you about genetics in LGS. And so I'm going to give kind of a broad overview, and I'll dive a little deeper into a few genes and some of the work that my lab has done, along with Dr. Ingrid Scheffer, who you probably heard. You'll hear some familiar things if you were at her talk yesterday, but it never hurts to hear things twice, I think. And tell you kind of what we know and maybe what we don't know, and next steps for continuing to learn about genetics and in LGS. So I'm going to talk to you a little bit about how we do gene discovery. So how do we determine that a particular gene is associated with LGS or other epilepsies? Talk about a few of the genes and give you some long lists of genes. So if you've had genetic testing, you might recognize something up there. And then kind of talk about how that fits into all of the other similar developmental and epileptic encephalopathies, which is kind of the umbrella term that we use now for epilepsies with a significant developmental component like LGS. So this is more to remind me, because all of you know, but thinking about how do we define LGS. And I think that's important when we do genetic studies. We need to remember when we find a gene, what exactly is it associated with, and what are the features of those kids with a change in that gene. So as you all know, there's some specific definitions for LGS that encompasses a lot of different things, including early onset seizures, developmental issues, multiple seizure types that you all deal with on a daily basis. I will also say my talk is not going to go for the entire 45 minutes. If you have questions along the way, I'm very happy to answer them, but I'll have plenty of time at the end. I think the important thing about the definition of LGS is that, as you all know, when your child or your loved one has that first seizure, you don't get a diagnosis of LGS at that first seizure. It takes time. And the phenotype or the clinical features evolve over time. And importantly, they overlap with many other epilepsy syndromes that also have multiple seizure types and a developmental component. And this is important for us in genetics too, when we think again about how are genes related to different types of epilepsy syndromes. So you may have had different diagnoses along your journey as well, and overlapping syndromes that I think are important include myoclonic atonic epilepsy, where again there's a significant component of those atonic or drop seizures. Dravet syndrome, this is one of the most well-recognized, but not always is it recognized. And sometimes adults with Dravet syndrome might get a diagnosis of LGS. West syndrome, as you know, can evolve to LGS. And there are other syndromes as well that fall into this overlapping category. And LGS can be misdiagnosed along the way or even for a long time as one of these other syndromes, about a third of the time. So as you all know, there can be many different paths to come to a diagnosis of LGS, including birth injury, stroke, brain malformations, infections, but about up to a third of patients may have a genetic etiology, and that's what our group is working on trying to understand. If we can understand the genetic causes of LGS, it'll help us understand perhaps where we can intervene, not only in the kids with those genetic changes, but perhaps more broadly across LGS. So I think it's important to stop and think about, why do we care about genetics? I'm very biased because I'm a geneticist, but I think genetics plays a really important role in the clinic. And I see patients in the clinic as well at Seattle Children's Hospital, not just epilepsy patients, but many different types of pediatric disorders. And understanding the genetic cause of a disorder is helpful for many different reasons. First of all, it might end that diagnostic odyssey that you may have been on, trying to figure out what is going on with my child or my loved one. And when we identify a genetic cause, we can ask who are the other patients that have been reported, and we can help understand then paint a picture of what life may look like for your child by using that information for other kids that have been reported. We can talk about how likely is it that this will happen again if I have another child, so recurrence risk, once we know the genetic cause. When we don't know the genetic cause, that's harder to do. In some cases, and increasingly, the understanding the genetic cause might affect the medications that you choose to treat epilepsy, and importantly, it might affect choice of medications to avoid that might make things worse. So this is becoming, I think, increasingly important in epilepsy. It's still rare. There's almost no magic bullet for any genetic cause of epilepsy, but we're getting better. And then I think finally, importantly, because we don't always have that magic bullet, identifying the genetic cause can actually give you opportunities then to participate in additional research studies where we're trying to find new treatments for specific genetic types of epilepsy. And if you don't know you have that genetic change, you can't participate in those studies. So thinking long-term about precision medicine opportunities. And then finally, and I think you saw a similar slide from Dr. Sheffer yesterday, identifying the genetic cause allows you to connect to other families who have the same genetic cause as your child. And this can be very powerful, just as this meeting is very powerful for families to connect with other kids and families affected by LGS. And when you have a specific genetic cause, you can then start to hone in on what types of things can we do. Are there specific things that my child has that other genetic types of epilepsy don't? And can we start to engage with researchers who are working on the gene for my kid's epilepsy? Yeah, so that's kind of my why do we care about genetics. Genetics is important for many reasons. But I just want to make sure, as I start to talk now about genes and how we identify epilepsy genes, I'm going to put us all on the same page. Genetics is complicated and it can be scary, but it's really not. All of us in this room, all of our cells contain DNA, and that DNA is packaged into these little, what look like X-shaped things here. That's what they look like under a microscope, sort of. And these are called chromosomes. So all of our DNA is packaged into chromosomes, and that's what we inherit from our parents. And as you read along that DNA, every so often, there's a gene. So each of those little red dots is a gene. And the genes are really the instruction units for building our bodies, for keeping our bodies functioning in the proper way, for the cells talking to each other. And when I say we're going to sequence the gene, we're just going to check the spelling of that gene, okay? And the spelling is something like this down here. It makes absolutely no sense unless you pull out that little Gattaca there, which I highlighted. So the movie Gattaca was based on, the name was based on the letters used in DNA. And importantly, we have 20,000 of these genes packaged into every single cell. And so it's our job as geneticists to figure out which ones are important for the development of epilepsy, okay? And the ability to check the spelling of the genes in our DNA has increased greatly over the last 10 years, and this is why we are now able to give a genetic diagnosis to about 40 to 50% of kids who present with an early onset epilepsy. I think Tracy showed a slide similar to this on the first day, kind of how genetics has exploded. So in 25 years ago, we had maybe a handful of genes that we were able to test in kids who had severe epilepsies. And so the ability of us to diagnose someone who walked into the clinic was maybe a few % at best of children or families affected by epilepsy. And as our technologies got better, and especially over the last 8 to 10 years, we have now been able to associate over 100 different genes with severe early onset childhood epilepsies. And each of these little, what look like words here, represents a gene that's been identified as associated with epilepsy over the past 8 to 10 years. And one of the reasons that we've been able to do this is that technology allows us to sequence lots of genes at once in you, in your child, and in both parents, and compare the DNA of your child to many other children and to you as their parents and identify new changes in the child. And those are called I'll refer to those as de novo or new mutations in the child. And most epilepsies like LGS are due to new genetic changes that neither mom nor dad has in their DNA, but is present in the child, and that's what causes their epilepsy. And so the technologies that we use, and I'll tell you about how we do that, and in the research lab, are also used in the clinic. So if you've had genetic testing, or if your child's had genetic testing, they've probably either had what's referred to a gene panel, or maybe even a whole exome, we'll talk about that in a minute, that allows us to sequence many or all the genes at once. So we've been doing gene discovery in epilepsy for almost 10 years now in our lab in Seattle. And most of this has been done in collaboration with Dr. Sheffer, who's here, who has been studying and collecting DNA from kids for probably twice that long, correct? So we met about 10 years ago and realized that that my lab had the ability to perform lots of genetic testing, and that Dr. Sheffer and her colleagues had a large number of patients who had early onset childhood epilepsies and wanted to understand if there's a genetic cause. And so we put those two things together and said, can we discover new genes for epilepsy? And so we've done this now for about 1,200 kids affected by an early onset epilepsy. Yeah, not all of those kids have LGS, as I show you here. So the individuals that we have been studying all have severe epilepsy, intractable seizures, and developmental delays, but they fall into different categories. And as I said, some of these are overlapping with LGS. And this is just to give you a sense for the different types of epilepsies that we've studied. And this is from one of our, about halfway through some of our studies, to show you that we had 46 individuals with LGS in our cohort, and we performed sequencing in about 50 genes at a time to try and find the changes that underlie that LGS. And we were able to provide a diagnosis to 5 of those families affected by LGS. And what I'm showing you here are the names of the genes that were the cause in each of those 5 children. And I think what's important here is that we had 5 diagnoses and we had 5 different genes. So each kid had a different genetic cause of their LGS. And so at this point, we had solved about 10% of the LGS kids in our cohort. I was involved in a large international consortium that was also interested in understanding the genetics of LGS and infantile spasms. And in that study, we looked at 115 kids with LGS, and we sequenced all 20,000 genes in the child and their parents to look for those new or de novo genetic changes that I told you about. And so here we were also able to solve about 20% of the kids with LGS and provide a genetic diagnosis to them. And here's a list of the genes in which we were able to identify the disease-causing mutation. And again, what you'll notice is that of those individuals, each gene is listed and was found in only one individual, unless I have that little times 2, times 3, or times 8, where we found multiple individuals with changes in the same gene. So again, we're starting to identify that LGS in some individuals has a genetic basis, but that when we take several different individuals with LGS that's genetic, almost every time those kids are going to have a change in a different gene. So this was a large international consortium I told you about. In our lab, we've done a very similar study. And this was actually started by Candace Myers, who some of you may have met on Friday, with funding from the LGS Foundation to look for genetic changes in kids with LGS. And the work is continuing by Dr. Allison Muir, who's here in the room today, who's been continuing to follow up on the results of this study. And again, we sequenced all 20,000 genes in about 200 kids with early onset epilepsy. And this little bar here are the individuals with LGS. And the purple at the top represents those kids that we were able to provide a genetic diagnosis for. So again, we were able to identify the cause in about 10% or so of cases of kids with LGS. And I'll tell you a little bit about some of the things that we found. So we had 18 individuals in this study who presented with LGS, and we were able to definitively provide a genetic diagnosis for 4 of them. And so there's a theme here, right? You'll recognize that these are the 4 genes in which we found a mutation, 1 in each child. And in 3 of those 4 cases, again, those were new genetic changes not present in either parent. Importantly, I think all of these were fairly recently discovered genes. So there are a handful of other kids in the literature who have a genetic change in this gene. Most of them have epilepsy. All of them have a developmental disorder of some sort, so developmental delay and intellectual disability. But their phenotypes are quite broad, meaning that they are all developmentally delayed, but they look a lot different from each other, some mild and some severe. But this puts, I think this makes sense. It makes sense to us in genetics, but this puts LGS in, we know, falls into a category of a broad spectrum of conditions, and all of them with similar features, but those similar features are a little bit diffuse, right? Developmental delay, seizures, intellectual disability. But we were able to provide a diagnosis for 4 of them, so that's about 20% of the kids. Importantly, in research, we can also look at things where we're not quite sure if it's the cause, and that's really the point of what we do, is to study things where we think, well, maybe this is giving us a clue to a new genetic cause of LGS. And so, for example, this is a list of about 10 genes where a child with LGS had a new genetic change in that gene. But it's never been reported before in the literature. We've never seen it in any of our other studies. But we think some of these might actually be the cause of LGS in these individuals. And so, for example, when we find this, then we reach out to others in the genetics and the neurology community and ask, have you ever seen a change in this gene before in a child with LGS or similar features? And there are many mechanisms for us to do that, including online databases, but also national meetings. And just as an example, I presented this list in December at the American Epilepsy Society meeting, and someone came up afterwards and said, I have a change in that gene in a kid with early onset seizures. And we've now collected about 6 individuals with changes in this gene who all have similar features. And so our hope is that I don't know, in the next few months, looking at Allison and Dr. Sheffer, that in the next few months, as we continue to collect the data, that we're able to put this out in a paper and say, here's a new cause of brain malformations in epilepsy. It just so happens that these kids also have brain malformations, but most of them have seizures that go along with that. Great question. So for those of you who couldn't hear it, Melanie's asking, I've met parents who say, my kid has a change in a gene, and people don't know what it means, not been reported before, but I met another parent, and their child has a change in the same gene. What do I do next? And I'll put some resources up at the end, but really what you need to do next, that's a really good question. It's contact someone like me or Dr. Sheffer. It can be difficult, but if you know another family with a genetic change in the same gene, there are online resources I'll show you at the end where you can actually enter your own information and say, my child has a change in this gene, and that becomes available to researchers like me, to clinicians, and to other families. There are some great examples of families who found each other online before physicians or researchers know anything about the gene. Another option is to use Facebook. So those of you who are on Facebook, there have been parents start Facebook groups to say, and just give it the name of the gene. It allows other people then to Google and find that, make that connection. But I would encourage you to reach out to, in fact, you could reach out to Tracy or to Melanie or to Christina as well, and they can connect you with people who might know what to do next and who to connect you with to really understand if that's the cause. Sound reasonable? Yeah. Yeah, so if you recognize any of these genes, please let us know. We're working on many of them. It does take time, so even when you find another parent and you come to us with all of your information, it's going to take us a few months at best to really sift through all of that and be able to present it to other researchers and to publish it in a form that's available to the public. But we are more than happy to do that. And I have absolutely published at least one, if not two, papers that started with a parent coming to us with information about their child and other affected children. Okay, so I'm just going to talk a little bit about 2 or 3 of the genes that we know about, just to give you a flavor of genes in LGS and how they are, how LGS fits into the spectrum. So CHD2 is actually one of the first genes that our group identified as associated with early onset epilepsy. And we found it I think 5 cases in 500 affected kids with early onset epilepsy. Of that, there were 6 kids with LGS, and one of those 6 kids had a change in CHD2. Since that time, there have been additional reports of CHD2 as a cause of LGS. So in the second study here, 2 of 22 individuals, and then in the third study I already told you about, 2 of the 115 individuals that we studied, this was their cause. Everyone with a change in CHD2 has developmental delay, sometimes autistic features, and almost always they have seizures. They don't always get a diagnosis of LGS, but they fall into that spectrum. This is a really recently described gene, HIVEP2, we're calling it. These were discovered by other researchers, but when we looked at our data, we found one child with LGS who had a new genetic change in HIVEP2. So this is an example where we then go back to the literature and say, what do the other kids look like who have changes in this gene? Does it make sense for our patient? And it does. But here's an example where only 30% of kids with changes in this gene have seizures, but they all have developmental issues, often severe. And there was one of the 9 patients presented in the literature, and one of our 18 patients have a diagnosis of LGS. And then finally, we recently identified this gene called CUX2, and we've diagnosed 9 patients so far. They all have exactly the same genetic change, and 2 of those 9 patients have a diagnosis of LGS, and the other 7 have severe developmental delay, multiple seizure types, and autism, but don't cleanly fit the criteria for LGS. They have one of those overlapping epilepsies. So this is a busy slide, but what I'm showing you here is the data from our studies over the last many years. And every purple bar here represents a gene, and the height of that bar represents the number of patients where we have found a diagnosis by a genetic change in that gene. So for example, the highest bar here is a gene called SCN1A. It's a sodium channel, and it causes Dravet syndrome. Oops, sorry. So as you can see, there are a lot of bars on this graph. And what that tells us is that there are many different genes in which you can have a mutation and have a developmental and epileptic encephalopathy. And layered on top of this, we see that the picture is very much the same for LGS. So every orange asterisk here represents a gene where at least one of the patients that we diagnosed had a diagnosis of LGS. So there are many different, not only are there many different ways you can get to LGS, from infection to trauma to genetics, within genetics, there are many different ways you can get to LGS by genetic changes in many different genes. This also tells us a little bit about the biology of developing epilepsy. And this is just a diagram of 2 brain cells talking to each other. This is where the action happens in the brain. And when things go wrong here at the synapse, it's called, the space in between, it gets too excited. This is when seizures happen. But what we know is that of the genes we discovered, they fall into many, many different classes or many different jobs that the cell has to do. And it means that compared to 20 years ago, we feel that epilepsy is much more complicated than we originally thought, where we thought one thing goes wrong at the synapse and you get epilepsy. Many different things can go wrong. But I think the opportunity here is that if you take all these bold headings here, these are all opportunities for us to think about how do we now develop precision therapies for kids with epilepsy due to mutations in these classes of genes. So every gene we identify, every group of genes that we identify, I think is a new opportunity to think about new treatments, because clearly that's a need in all of the early onset intractable epilepsies, including LGS. So we've diagnosed probably 10 to 20% of kids with LGS through our research studies. And I think my guess is that that's probably similar. Dr. Sheffer would have a better sense for genetic testing broadly in LGS. We can find a diagnosis a handful of percent of the time. And one of the goals of our lab is to say, well, what do we do next? Because there are many of you in this room, there are many in our studies who we think their epilepsy should be genetic, but we can't find the change. So just to remind you, earlier I said that as we walk along the chromosome, every once in a while we find a gene. And even when we sequence all 20,000 genes or instruction units in the cell, that represents only about 1% of the DNA in our cells. And so our next step is to ask, what about that other 99%? Can we find changes there that might be related to epilepsy? And so one of Allison, who's here in the room, one of her projects is to do this for patients where we don't have a genetic diagnosis, and to sequence every single letter of the DNA in our cells, or the whole genome, and try to identify changes that might be related to epilepsy. That's a little bit more complicated. It's a lot more data, as you can imagine, to sift through and to interpret. And so the other thing that we're doing, which I think is really cool, is collecting cells from patients, usually skin cells, so a little punch skin biopsy. And the reason we do that is that we can take those skin cells, we can convert them back to stem cells, where they can turn into any type of cell, and then we take those and we make brain cells, or even little three-dimensional kind of collections of neurons that act a little bit. They're not real brains, but they have some of the same types of cells. This allows us to take a skin biopsy rather than a brain biopsy, which is a lot more complicated, and try to study the genes that might cause epilepsy in our patients. This also allows us to do lots of different types of studies than just sequencing the DNA. And so Malavika, who's also here in the room, who works in my lab, is going to look at other types of changes that might be related to epilepsy that we wouldn't have found by the testing that we've done so far. So why do we do this? One of the goals, obviously, is that we really need to improve the ability to treat patients with LGS and other early onset epilepsies. The goal is precision therapy, so that if you identify the genetic cause, that will tell us what treatment to use. We can do this in a small, like I said, a small percentage of cases today. That percentage of cases is growing. So here are examples of recently identified genes where there are medications that may or may not work, but are being tried in some cases. And our hope is that we can move from 4 or 8 or 10 genes where we can provide this information to families to all 100 genes associated with epilepsy. If we find a genetic change, we can tell you what medications might work and might not work. So I think so we talk a lot about precision medicine. Yeah. Mm-hmm.
Then, and we know that that might be a good match, then do we also assume that there could be medications that are not good for that particular? Okay.
Yeah.
So then, how do clinical trials help us if we have people trying medications without knowing?
You're singing my song.
If a child has no genetic testing,
Yeah.
and they say, let's end this clinical trial.
Yeah.
And you have this huge adverse effect.
Great question. Yeah.
How it's like horse cart or whatever that is.
Yeah, great question. So for those of you who couldn't hear, the question was, the goal is to use the genetics to help us identify medications that may work or that may not work. How do we fit that into clinical trials, and how do we, I guess, when you're in clinical trials, how is that used, or is it? And for the most part, it's not today. It's moving in that direction. There have been some trials, for example, for Dravet syndrome, and Dravet syndrome is caused by a single gene, SCN1A, and most of the time those trials require that you have a genetic test that shows that that's the diagnosis. There are definitely clinical trials on where I think in the background they're doing some genetic testing to start to answer those questions, because it is a really important question. If you're doing a clinical trial, and I think these trials are important, right, because we need new medications whether or not you know the genetics. So there are trials that go on, and you have a clinical reason to enter the trial without knowing the genetics. But it is important. Some kids will do very poorly and some will do very well. And that might not be, but it might be because of their specific genetic cause. And so I do think it's really important. And I've actually talked to a few companies along the way and say, this is really important. You should have this information layered in, because down the road you may actually be able to, again, divide up those patients, not just by their type of epilepsy, but by their specific genetic type of epilepsy. Right, right. Yeah, it's hard. And like I said, for most of the genetic causes, we don't have that kind of fine-tuned information about what medications work and what don't. For a subset, we definitely do. So for SCN1A and for SCN8A and a few others, we definitely do. But for the most part, we don't. And I think it will be important down the road as these clinical trials move forward to try to layer in that information. It could be difficult because I know you talked yesterday about how do you get genetic testing, and genetic testing is not easy to get in many places and for many families. And so it has to be a part of and we can talk about that too, but it really should be, I think, a part of the trial, in my opinion. Great question. So I think we've gotten a lot better now about being able to provide a precise diagnosis for some families with LGS and other related epilepsies. And we're moving towards using that information to be able to provide precise therapies. I also think that genetics is showing us many different biological processes involved in epilepsy, and that, again, those give us new opportunities to think about how to intervene and treat or even prevent seizures. I want to make the point that research takes a long time, so if you're involved in research or you don't yet have a genetic diagnosis for your child, be patient and get involved. And I put our lab's information up at the top there. The question that was asked earlier by Melanie about what if we have, I know another family with the same gene, or how do I find another family with the same genetic cause? This resource here, MyGene2, is just one of many resources. This is actually, families can go in and enter information, as much information as they want about their child. You can put pictures, you can put their clinical history, you can write their story, and you can put their genetic information in. You can also just go in, sign up, and put their genetic information in if you don't want to share all of the information about your child. And this will automatically try to connect you with other individuals who have the same genetic cause.
Is that for adults too?
Oh, it's for anybody. And it's for actually, it's for any disorder. So it's not just epilepsy by any means, but it's, we put a lot of our research data in there that without, stories about the individuals, but with the genes, so that, again, it's another way for us to help identify other families. So LGS is genetically heterogeneous. That's a fancy way of saying there are many, many different genes that can cause LGS. And the quote-unquote LGS genes are going to be a subset of the genes that cause other similar types of epilepsy. What this means clinically is that if you do get genetic testing or are able to get genetic testing, we almost never test one gene at a time anymore. We test many genes at once because we know there are many different genes that can cause similar features, or we test all of them, and that's the exome sequencing that you probably heard about yesterday. And then I just wanted to end with the point that, Genetic testing is important, and I talked about how it can connect you to specific organizations or family groups based on the genetics. What I want to emphasize is this doesn't take away the diagnosis of LGS. This just provides you another entry point into connecting with families and individuals, and that it will give you 2 homes instead of one here at the LGS Foundation. But I think they're not mutually exclusive, and that many of these genetic epilepsies also have LGS as part of their diagnosis. And it's perfectly fine to be involved in more than one organization and try to help us move the research forward and help the other families who are affected in the same way. So I'm going to stop there. This is some of my group. We've been very fortunate to have funding from the LGS Foundation for some of the work that I showed you, as well as from many other different funding organizations. And I have to thank Dr. Sheffer, Dr. Muir, and Dr. Hebar, who were in the room and did some of the work as well. So thanks.